I'm here at the EXIT International Conference because my lab's interested to understand how bacteria called Staphylococci escape from macrophages that have taken them up.
Now that I have seen so many of the talks, it's a program like this, they create this sort of critical mass of people that every single lab could not achieve.
At least every intracellular pathogen needs to exit, and to do this to take this as a main theme for infection research in Germany, I think is likely to come up with a general, or at least some general solutions.
Hello, I'm Alex Weber from the University of Tübingen.
Hi, my name is Markus Meissner. I am the chair for experimental parasitology at the Ludwig-Maximilian University in Munich.
I'm Ute Distler and I'm managing the co-facility for mass spectrometry in the University Medical Center in Mainz.
I'm Uli Doren. I'm a microbiologist working in Münster Institute of Hygiene.
So I'm Laura Bindila. I'm head of the clinical immunologics unit at the University of Mainz.
Hello, my name is Michael Hensel. I'm heading the division of microbiology at the University of Osnabrück.
My name is Jonathan Neuber and I am a PhD student at the Robert Koch Institute in Wernigerode.
My name is Alfonso Felipe Lopez. I'm from Mexico. I am a scientist at the group of Professor Anja Lührmann and the University Clinic in Erlangen.
I work with Legionella pneumophila. Particularly, I focus on the secreted phospholipases and their role on the lysis of the whole cell. So when Legionella wants to escape from the cell and trying to infect new cells.
Here we work with Toxoplasma. It's the cat parasite. It's worldwide distribution. Best known probably because congenital toxoplasmosis still causes a lot of aborts.
And we are studying the strategies to understand how Coxiella burnetii, the causal agent of the Q fever, is escaping from the cell.
And in this priority program I have actually two projects. One is our main pathogen Salmonella enterica, where we look at escape from the Salmonella containing vacuole. And the second project I'm heading together with Katarina Psataki, and this is a project providing an imaging platform for various members of this platform, this priority program. So this is the idea to give everybody access to high resolution imaging, correlative microscopy, ultrastructural analysis.
We do focus on protein analytics and have instrumental platforms which allows us to detect novel or find novel proteins, differentially regulated proteins, to distinguish maybe healthy samples from diseased samples. Or the outcome that we can achieve with this technology is quite amazing, because we could also monitor protein expression profiles. For example, if you infect a cell, a host cell, we can maybe see which proteins change in that cells, which pathogenic proteins are expressed in the cell.
Lipid and lipid homeostasis and dysregulation is extremely important in infectious disease. In fact, a lot of people are trying to medicate themselves and therapy themselves using different, lipid or fat-based therapies against infection, like anti-inflammatory fat molecules. So just through this connection, maybe for the public, it's clear how important the lipids are in treating the infections, but also in the mechanism of infection, right? In initiation, progress, and also in the spread of infection. So at the cell membrane level, basically every cell membrane will modify its lipid content in order to accommodate or allow pathogens to infect intracellularly or to escape and replicate further. So therefore, lipid very important.
We have so many different microbes. I think it's important to understand how each of them interacts with the host and how there are sort of special ways of immune escape or of killing the host cells. So I think it's important to conduct that research.
Where bacteria, fungi, viruses really try to interact with those cells, hide so that the immune system cannot really catch them, that you cannot treat them with antibiotics or antifungals or any kind of medicine, then it's really cool to invest money in order to really spot them in these places and see how they get out of the cells and how they find their niche, their way to escape and then start a new of infection. If you want to understand how pathogens hide somewhere and then all of a sudden get out of their intracellular reservoir, then we have an idea how we can really treat them. Just consider all these chronic infections.
During the first funding period, we identified 2 new crucial factors. They are only present in the genome of apicomplexans. And the hope is, of course, if we can now develop drugs, that we can block the host cell exit not only of toxoplasma, but also of malaria and potentially other apicomplexans.
If we understand how the bacteria escape from the cell, then we can understand how disease is developing within the body. And then we can understand how the bacteria not only infect some kinds of organs or in a localized infection, but also to understand how other organs within the body get ill by the bacteria. For example, with the hair, in this case, the Q fever and Coxiella burnetii also appear in the hair, causing endocarditis, and this is one of the reasons we are trying to understand this mechanism of escape from the host cell.
And we know a lot about the entry of Salmonella, so invasion processes are well studied. Also how Salmonella can replicate inside host cells is well investigated, but the last step, how to get out of a cell, be able to reinfect and spread the infection in a host, is not that well understood. And this we hope to unravel, and our specific focus is this event of getting out of a vacuolar compartment to be able to spread from uninfected cell to other cells.
Well, first of all, we're dealing with a lot of pathogens that are really important, disease-wise. And also, as a fundamentalist, you're also sort of discovering very important cellular processes that can be applied to other pathology. But I mean, it's no question that, disease-wise, it's really important, and it's good that this program allowed to address these points, and I'm sure from what I've seen, you're really generating top-notch data. So you see there's a link between infection and Parkinson's disease, because the Parkinson's disease protein, they regulate the way a cell will react to a stress caused by infection. But on the other hand, the infectious agent will manipulate the cell to sort of counteract. So I think there's a lot to learn from this sort of the 2 angles, even for Parkinson's disease, So that's, for me, that's quite interesting.
The 12 million are really well spent money, because it really prevents people from being too specialized into their own niche, and it really forces people working on a variety of very relevant pathogens to come up with generalizable solutions.
This priority program really capitalizes on bringing different people together. So I think we need the program to learn from each other, to bring our expertise together, and in that way make real progress. So I think that's the key thing.
Obviously, this research involves very expensive equipment. These parasites are very small. And so we need the necessary funding to basically do this sort of very basic research at this point, but we hope to translate that in the future to intervention strategies.
I mean, without the funding, I couldn't. It's extremely important because you need people to do the work, and we are relying on PhD students, and the PhD, of course, have to be paid. And we have a lot of enthusiastic students, and they need at least 4 years to ask these questions and to get meaningful results. You need at least 4 years, and you need funding.
The question about the amount of funding is relative. I have family members who work in aeronautics, and if you talk to them and they talk about the amount of money they need to build a new airplane, it's 1 log higher than what we actually need for infection research. I think through the SARS-CoV-2 crisis, everybody should have realized that research on infection is important. And I still believe that the funding amount in comparison to other research that I mentioned is actually still very little.
You might overlook so many things if you do not really search for it. So therefore, I think it's really mandatory to take this money in order to search at the right place and also find out how they can escape and exit. So that's a cool topic.